Packed inside every cell in our body is a set of genetic instructions 3.2 billion base pairs long. Deciphering these directions will be a monumental task but could offer unprecedented insight into the human body. Accordingly, in this video, we will learn about the biomolecular technique that paved the way for unlocking the secrets of life encrypted in our DNA. The Sanger sequencing. Before we begin, let us first have a short recall about the central element of our discussion, the DNA. Deoxyribonucleic acids are biomolecules consisting of long chains of nucleotides. These nucleotides can be adenine, thymine, guanine, or cytosine, and can be arranged in different combinations and orders. The DNA and its sequence serves as the blueprint of life, dictating how an organism looks and behaves. Basically, who we are. Let's take a look at the gene that codes for hemoglobin subunits, the hemoglobin beta gene. This protein subunit is essential in making hemoglobin, the proteins that enable our red blood cells to carry and transport oxygen throughout our bodies. The image on the screen shows the exact order of nucleotides that made up this gene. Pretty lengthy, right? So, how did we figure this out? How did we uncover the exact order of these nucleotides, ATG, GTG, CAC, and so on? Correct. One of the pioneering and reliable technologies that enable us to decipher these codes is the Sanger sequencing technology developed by Frederick Sanger in 1977. Sanger sequencing is a biomolecular technique that allows us to determine the exact order of nucleotides in a DNA molecule based on polymerase chain reaction and the random incorporation of chain-terminating nucleotides called dideoxynucleotide triphosphates or DDNTPs. To execute a Sanger sequencing, first, a polymerase chain reaction is performed to specify and amplify the desired DNA sequence. In this process, the reaction mixture is heated up to 95 degrees Celsius to denature double-stranded DNA into single-stranded ones. After that, the temperature is reduced to about 55 degrees Celsius, allowing the primers to bind with its complementary ssDNA sequence. The temperature is then increased again to about 68 degrees Celsius to enable DNA polymerase binding for DNA elongation, wherein nucleotides are added one by one. Eventually, DNA elongation is terminated upon the random binding of fluorescently labeled DDNTPs. These modified DNTPs do not have the alcohol group in the 3' carbon of the deoxyribose necessary for DNA polymerase and nucleotide bindings. The process of heating and cooling the mixture is repeated for up to 30 cycles using a thermal cycler. This process generates considerable copies of target DNA having different lengths due to the random incorporation of fluorescently labeled DDNTPs. All these DNA sequences of varying lengths end with a DDNTP molecule. So, what do we do now with these differentially cut DNA fragments? Here comes another molecular technique, capillary electrophoresis. After performing a chain termination PCR, we use capillary electrophoresis to analyze and interpret the differentially cut DNA fragments and read the exact sequence of nucleotides in the target DNA. In this method, DNA fragments are transferred into a capillary tube filled with a porous separating medium submerged in a conductive buffer. An electric field is then applied to the capillary tube to drive the migration of DNA fragments from the anode to the cathode. This arrangement facilitates the differential separation of DNA fragments based on size and charge, wherein shorter and more negatively charged DNA fragments travel faster in the medium. A laser at the end of the capillary tube excites the fluorescent label in the DDNTP of each DNA fragment, resulting in the emission of specific wavelengths or color of light depending on the type of DDNTP, whether it is DDATP, DDTTP, DDGTP, and DDCTP. These emitted lights are detected and recorded by a highly sensitive detector and interpreted into a chromatogram. The image presented shows an example of a chromatogram reading with peaks of different colors. Each peak in this reading represents a DNA base. And the order of these peaks depicts the sequence of our DNA target. Amazing, right? This technology plays a crucial role in unlocking the fundamental basis of diseases by enabling researchers to identify mutations in genes that might be responsible for specific diseases. By comparing the DNA sequence of healthy individuals to those with a particular disease, scientists can pinpoint variations that could disrupt normal gene function and contribute to the development of the disease. In addition, Sanger sequencing also plays a role in targeted DNA analysis, such as DNA barcoding, a technique that allows us to distinguish accurately between different organisms. That's all for today, folks. I hope you learned something cool and important today. And remember, the mind is not a vessel to be filled, but a fire to be kindled. Till next time. Thank you.